In so doing, we're transferring energy in the form of building a very large proton gradient in the intermembrane space, not the inner membrane space. I saw that once. I saw that a lot of you, I don't know if you read the question carefully or you did not, but a lot of you sat there and thought that somehow the membrane of the mitochondria was the cell membrane. And I'm not sure how that happened, but please make sure you understand that the mitochondria is within the cell, and the membrane of the mitochondria is certainly not part of the cell membrane. So the question posed the following, that is, what would happen to the production of ATP if you came along and you increase the concentration of potassium in the matrix? And this is something that all of you should know, is that the movement of ions is dependent upon its electrochemical gradient. We talked about that up in the lab, well before we got to the exam. And by loading up the matrix, what happens to the electrochemical gradient that's driving protons through the pump, or making that pump run backwards? Is that electrochemical gradient going to be the same? Is it going to be larger, or is it going to be smaller? And the answer is, it's going to be smaller. And therefore, the production of ATP will go down. Also, there is a question about the alveoli and the effect that COVID-19 had on the lungs, including the development of pneumonia, where you get this accumulation of fluids within the alveoli, and also between the alveoli and the pulmonary capillaries. Many of you did not even talk about the Fick equation. Anytime we're talking about diffusion, you have to default. You got to mention, you got to sit there and say, here's the reason why I'm saying what I'm saying. And it all revolves around the Fick equation. Many of you recognized, just as we had discussed in class, that the accumulation of fluid within the alveoli basically functionally reduces the surface area, the diffusive surface area. And of course, by there being an accumulation of fluid between the alveoli and the pulmonary capillaries, that's increasing the diffusion distance. But I want to encourage you always to go to fundamental principles that you know, and hopefully those will help you solve questions that I haven't directly addressed within class, but I have given you the tools to try to answer those. And the same thing applies to things like the Nernst equation. The equilibrium potential, what the heck is an equilibrium potential? That's where those rules of electrophysiology will help you out a lot in thinking about what we're going to move into in lab tonight. Okay, so that's my little mantra for the night. So here we are talking about cell-to-cell communication. We talked about autocrine, paracrine, neuroendocrine, endocrine type systems. I gave you a very generic sketch of what that entails or means. And then the point that I try to make is that in talking about autocrine, paracrine, neuroendocrine, endocrine type systems, there are these cells that are releasing chemical messengers that are going to go convey information. They're going to go talk to these target cells. And so that was the only description I provided. But then I follow that up. I started saying, okay, so how is it that the chemical messenger actually acts on the target cell? And that gets us into what I call the 4 different types of transduction. So let me define for you what transduction is. I did not do that on Monday night. So please write this in your notes. Transduction is the process by which an external signal produces an intracellular response. It's simple. So transduction, the process by which an external or extracellular signal produces some type of intracellular response. Now in the past I've had students sit there and say, oh, the extracellular signal is being converted into the intracellular response. No, I'm not saying that at all. I'm saying that there is this chemical messenger, the external messenger, that ends up through a series of events producing an intracellular response. So don't sit there and tell me that, oh, the extracellular response turns into the intracellular response. Are you guys with me on that? Glucagon, the hormone that's released from hepatocytes, it binds to a receptor. That's the extracellular response. That's the chemical messenger. But glucagon is not converted into anything inside the cell. It just binds to a receptor and creates a cascade of events. What do you guys think? Is that clear? So 4 different types of transduction. What are those 4 different types of transduction? Second messenger systems, receptor-gated ion channels, kinases, nuclear hormone receptors. As it turns out, there's 4 different types of second messenger systems. What are those? Cyclic AMP, cyclic GMP, calcium, and so-called IP3 or inositol triphosphate, second messenger systems. So if there's a question that says, name the 4 different categories or types of transduction, you're going to sit there and say second messenger systems, receptor-gated ion channels, kinases, nuclear hormone receptors. If there's a question that says name the different types of second messenger systems, you're going to sit there and say cyclic AMP second messenger systems, cyclic GMP, calcium, IP3. Is that clear? All right. Well, so tonight what we're going to do is we're going to continue on. I'm going to start by talking about calcium as a second messenger system. And then from there, that will take you to talking about inositol triphosphate, otherwise referred to as IP3. You need to be very cautious in understanding that IP3 is going to act not on the cell membrane, but on the membrane of a organelle. The mitochondria is an organelle, is referred to as an organelle. Organelles are basically these small subcellular structures. And hopefully, once I'm done with that, which I probably won't get through tonight, but we'll see, we'll get into an overview of hormonal regulation, and I'll provide a summary table. And typically, Stephen back there can tell you, every time there's a table, somehow, some way, those things show up on the test. And then we'll get into a very important aspect of homeostasis, and that's these so-called negative feedback loops. The negative feedback loops are at the heart of the cells being able to control their intracellular environment. Any questions? Don't be bashful, speak up, please. Anybody wants to talk to me about their test results, I'm happy to do it immediately after class tonight, after lab tonight, or you can email me, and we can set up some type of a Zoom call to go over the information. Some of you might sit there and on a question you got 10. It was worth 10 points and you got 9. I like it when the good students come to me who got 99.99% points go, hey, what, they're really big. Hey, why did I miss this 1 point right here? If that's you, don't worry about it. 1 point's not going to make any difference about how you do in this class. Now, if you're confused in terms of what you missed, I'm happy to explain that to you, but don't worry about that. I'm more concerned about individuals who missed large chunks of test questions. Let's see here. Okay. All right. So here's the scheme I left with on Monday night, talking about cyclic AMP as a second messenger system. And typically the general outline to this is that there's a first messenger, there's a receptor, here's our receptor, there's a transducer. Remember, this is all about transduction. there's a transducer. The transducer is basically going to convey the information that's arrived at the level of the receptor and then act on the amplifier. In talking about cyclic AMP, the amplifier is, you'll see some people say adenylyl cyclase, others will say adenylate cyclase. That's going to convert ATP into a molecule known as cyclic adenosine monophosphate, AMP. That's our second messenger. And as cyclic AMP levels go up, we convert a protein kinase that's in its inactive form. That's where there's a capital I here. This just represents a protein kinase that's in its inactive form, and the presence of cyclic AMP causes this inactive form to be converted into the active form. And all of you would tell me, hopefully tonight, that kinases phosphorylate their target molecules, which in most instances are other proteins. And let me just emphasize, and I mentioned this before, that by phosphorylating proteins, we alter their structure, and that alters their function. And a ton of our physiology is built around that. Let's see, what else should I mention up here? I think that's good enough. And I gave you an example of something like this, and I used glucagon as an example. But there are many, many, many other examples. And as we get into the cardiovascular system, I'll begin to provide many more specifics about what's going on further down here in this scheme. Okay, now, with regards to calcium as a second messenger system, here's the calcium ion channel, and we'll say that the calcium ion channel is closed. There's some type of stimulus that arrives at the cell and causes this calcium ion channel to open up. And when that happens, you'll tell me that calcium is going to move into the cell according to its electrochemical gradient. Our cell is at minus 70 millivolts. Test question time. Is the electrical chemical gradient large or small for calcium?
Large.
It is large. You guys all learned that its equilibrium potential is 480 millivolts, right? I guess that's right, 480 millivolts. Let's see here. Anybody wearing imaginary bullseyes tonight? She is? Is that what you said? Were you pointing at her?
Yes, absolutely.
That's what I thought. I don't want you to answer. I'm going to pick on, I'm going to ask somebody else. Where's Jocelyn? Hello, Jocelyn. Can you tell me what is the equilibrium potential for calcium? You can look at your nose.
It's 120 millivolts.
120 millivolts, that's exactly right, very good. So the cell's at minus 70 millivolts. We've got this huge concentration gradient for calcium, right? It's a 10,000-fold concentration gradient. And then on top of that, we have this very strong electrical gradient that also wants to drop calcium ions into the cell. And also, just to put a punctuation mark after this story of calcium, remember one of the points that I made to you is that if we were all talking in here at the same time, we would have a hard time understanding what any individual in here is saying. That's called noise. We'll say it's just a lot of noise. On the other hand, if it's really quiet in here, then I can speak with a very, very low voice and you guys can hear me. For the cell, that noise is all the things going on inside the cell. But here comes along calcium. And remember, one of the points I made to you is that the concentration of calcium inside the cell is very, very low. And therefore, any small amount of calcium that comes into the cell can cause fairly significant intracellular responses. So this idea that calcium has a very, very low concentration inside the cell is incredibly important for the cell to be able to detect the signal. Everybody with me on that one? All right, so calcium comes into the cell, and when it does that, it comes in contact with this calcium binding protein. And the name of this calcium binding protein is calmodulin. That seems to me like a good name. If you look at that, for me, when I look at that, I'm sitting there thinking, oh, cal, that stands for calcium. And modulin, to modulate, to regulate. So this calmodulin is allowing the system to regulate itself based upon the presence of calcium, calmodulin. I'm going to simply use capital C, lowercase m, capital D to represent calmodulin. Calmodulin is a calcium binding protein. And you're going to find out that these calcium binding proteins are really important. Where's my George Brooks student in here? Somebody in their test was talking about George Brooks. Nobody's going to admit that that was you, huh? Did you take a class from George?
No,
You're just looking at his YouTube videos?
yeah, or like podcasts.
His podcast. Good. Well, George is up at the University of California, Berkeley, and he's a colleague of mine. I've known him for 40 some odd years. For him and for his podcast, what turns on a muscle? Let me ask you a different question. What causes a neuron to release its neurotransmitter? It does. I showed you that, right? Didn't I show that to all of you last week? What's the signal that turns on our muscles? Calcium. I don't know. Calcium seems pretty darn important to me. All right, so calcium comes into the cell and it binds to calmodulin. Now, I mentioned this before. I'll just say it again. Whenever you see me writing a plus, that means that things are dissociated. So for instance, you see me write ADP plus PI. That means that the ADP and the phosphate that came off of it are floating around, not attached, not bound to one another, and completely independent of one another. So that plus, you're gonna interpret to mean dissociated. And when I use an asterisk, you're going to understand that that means bound to. So here I'm going to write calcium, and I'm going to put asterisk CMD. Just to be clear, there's lots of calmodulin floating around in the cytoplasm of the cell, moving around randomly, and now all of a sudden here comes a little bit of calcium, and through random molecular motion, the calmodulin comes in contact with it, and it binds that calcium. And when this happens, this causes a protein kinase that's in the inactive state to alter its structure into the active form of this protein kinase. And without going any further, you're going to tell me that somehow, some way, this protein kinase is going to create an intracellular response. Stop there for a moment. Any questions, confusion about what I've mentioned? Don't be bashful, just speak up.
So the calcium will, wait, no. The calmodulin will bind to the calcium once it enters into the cell,
That's correct.
right?
Uh-huh. That
And
changes the
that
structure
changes
of the
structure
calmodulin.
of the calmodulin, and it makes what's IPK?
The inactive protein
The
kinase. It doesn't
okay.
make it. It
Or
takes
it-
a protein kinase
and
that's
then
in an
it
inactive
activates
state
it
and turns it on.
and turns it on. And we're not going to get into the specifics of what
Not yet.
turning on is.
That's
Okay.
right. We will later. All right. Okay, now we're going to talk about inositol triphosphate as a second messenger. And the abbreviation for this is simply IP3. Inositol triphosphate. Oop. So here's a receptor. Here's the first messenger. Here's our receptor. And here's this protein. I'm just going to put PLC. Don't know how well you can see that in the back or even up front. PLC. And this stands for phospholipase C. And when I look at that big fancy word over there, phospholipase, what I really see is I see an enzyme that acts on phospholipids, right? Because phospholipid lipase, phospholipases are acting on their target molecules, which are phospholipids. And here's a phospholipid right here. Or you could say up here. So when our first messenger comes along and binds to the receptor, that changes the structure of that receptor. Ultimately, that then comes along and changes the structure of the phospholipase and basically causes the phospholipase to alter its structure from a off state to an on state. And one type of a lipid would be known as inositol diphosphate. IP2. And so phospholipase C, what it does is it comes along and it acts on this IP2 and converts it into a product, IP3, inositol triphosphate, plus. Let's see here. Eric, you're the bat. There are different types of fatty acids. Do you know what those categories are?
Saturated or not saturated?
Okay. And there's different types of acylglycerols. What are those?
Mono, di, tri?
Mono what?
Di and tri?
Mono, di, and tri what?
Glycerides?
Mono acylglycerols,
Uh-huh.
diacylglycerols, and triacylglycerols. Right? Yeah? Well, we're going to take our inositol diphosphate and we're going to convert it into two products. One is IP3, and the other, I'm just going to write up here, DG. And that stands for diacylglycerol. And diacylglycerol is it a protein, is it an amino acid, is it a carbohydrate, what is it?
Carbohydrate.
Okay? You guys can tell me that it's a what? Starts with an F, ends with a T.
Fat.
It's a fat, it's a lipid. And so, since it is, that stays membrane bound. It can't come off and go floating out into the extracellular space, nor can it go into the intracellular space. So it stays membrane bound. Okay, what's going on with IP3? Here's an organelle, and this organelle is known as the endoplasmic reticulum. I'm just going to call it ER. Endoplasmic reticulum. Let's see. Oh yeah, Ben. Ben, what's the concentration of calcium outside the cell?
Ben or Bennett?
Ben, well, Ben.
Okay. I just
Did
wanna
you-
make
you
sure
go
there's
by
no-
Bennett?
no one else. Yeah, I usually do. I just wanna make sure there's no one
Okay,
else. with
you
Ben.
tell me which one to be called?
Bennett's fine. No, Bennett's fine.
All right.
What was the question? Sorry.
What's the concentration of calcium outside the cell?
It is 1 millimolar.
That's correct. Good job. What's the concentration of calcium here in the cytoplasm?
1 micromolar.
Beep, beep, beep. You know, I started doing my little beep thing, you know, that,
Nanomolar? I don't remember if it was
Micro-
oh my goodness. Beep.
Mic-
Look,
micromolar?
listen, feel, no pulse, begin CPR. What is it?
1 micromolar. 0.1 micromolar.
Beep. beep, 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 beep. Yes, that's right, 0.1 micromolar. So the concentration of calcium inside the cell is 0.1 micromolar. The concentration of calcium here inside the endoplasmic reticulum is about 500 micromolar. That's 5,000 times greater than it is in the cytoplasm. It's about half of what it is outside the cell. Located on the membrane of the endoplasmic reticulum is a receptor-gated calcium ion channel. Say that again, receptor-gated calcium ion channel. Here I'm just going to write R period receptor, G gated, Ca for calcium, and then I period C period. You're going to read that as a receptor-gated calcium ion channel. And that receptor-gated calcium ion channel has two main components. Number one, it has a calcium ion channel. But the open-closed state of that calcium ion channel is dependent or predicated upon the receptor, the ligand binding site of that receptor being occupied. And in this case, what's the ligand that binds to this? And the answer is it's IP3. And when that happens, when the ligand binds to the binding site of this receptor-gated calcium ion channel, it changes the structure of the receptor-gated calcium ion channel, causing that calcium ion channel to go from being closed to being open. And you're going to tell me that calcium will flow out of the endoplasmic reticulum according to its electrochemical gradient. Where's Andrew? Andrew, you're up to bat. What's going to happen next? The answer's on the board, is it not?
Oh.
Concentration inside of the cell to decrease dramatically.
Okay. Anybody else going to help them out? Kaylee?
Oh no.
Oh, yes.
Oh no. So
The answer's on the
calcium
board, it's staring all of you right in the face.
is it that it binds with the CMD? No?
It's going to bind to cal
The
modulin,
cal modulin, yeah. And then it'll turn on it'll activate a protein kinase.
There you go. Good.
I did it.
And it's going to create some type of intracellular response to be defined in more detail downstream in class. So oftentimes what you'll hear or read is that the chemical messenger is the first messenger, which I've got up there. IP3 is referred to as the second messenger. And sometimes you'll see that in this story, calcium is referred to as a third chemical messenger. Okay, so going forward, you need to know how it is that the second messenger systems involving cyclic AMP, calcium, and IP3 operate. Become very, very important when we get to the cardiovascular section. And also, I will say also to the respiratory section of the class. Without anybody divulging their health, there probably are some of you in here who are asthmatic. And if you sit there, if you know somebody who's an asthmatic and they're having some tightness in their chest, or they feel like they're having shortness of breath, they take this little device. Some people call it a rescue inhaler, some people call it a nebulizer. And what they'll do is they will take a couple puffs. This device will basically produce this gaseous air that they will breathe into the lungs. And within that nebulizer, within that rescue inhaler, there's a drug known as what? Albuterol. And albuterol is going to act on one of these different mechanisms up here. And all of a sudden, several minutes later, you ask the person, how are you doing? Are you able to breathe easier? Oh yeah, I can breathe a lot easier now, no problem. And that's the cool thing about pharmacology. A lot of the pharmacology lies within the schemes that I'm laying out here for you. All right, moving forward. Just give me 1 second here. That's good. All right, so I'm going to come along and start down over here. I'm going to erase this part of the diagram. And now I'm going to talk about kinases. And specifically, I want to talk about tyrosine kinases. So, Mr. Corey, you're going to tell us, what is tyrosine? That's a carbohydrate, right?
No.
No? It's a lipid? It's a fatty acid? No? It's,
Nucleic acid.
it's a nucleic acid. Okay? Anybody else can help me out? Is it an amino acid, maybe?
Are we thinking of the same thing?
Tyrosine, amino acid. That name tells you exactly what the target is of the phosphate, the kinase, is to phosphorylate a tyrosine residue. In other words, a molecule of tyrosine within a protein. So here is a tyrosine kinase. And these are proteins. These proteins can have different functional regions. Sometimes people say domains. So make sure you write that down, is that these proteins are very, very complicated structures. They're not simple structures by any stretch of imagination. And so they can have different domains or different regions. And for tyrosine kinases, they have alpha subunits. And the alpha subunits actually serve as the receptor portion of this kinase. So there they are. They're in contact with the extracellular fluid. So any chemical messenger that is specific to these binding sites will bind to it and then cause something to happen. There also are beta subunits. These are all part of the same molecule. I'm just showing you these little dash lines here just to show that they're chemically bound to one another. So what I want you to start off with is an appreciation that the alpha subunits are the receptor part of this molecule. The beta subunit is the business side of this company, so to speak. This is what's going to cause intracellular response. I'm just going to put this little purple cartoon in the back. It probably looks like a black line, but it is purple. And beneath that, I'm going to write GLUT4, GLUT4. Where do we find GLUT4? What type of cells? Skeletal muscle cells, that's exactly right. Here's a vesicle. Okay, within the membrane of this vesicle are GLUT4 glucose transporters. And I'm just going to write here, inside the circle, I'm going to write GLUT. Now I'm just going to put VES. And you're going to interpret that to mean that this is a GLUT, in this case, a GLUT4 vesicle. This is a vesicle that has a bunch of GLUT4 transporters within its membrane. Remember, the fundamental structure of these vesicles, these are lipid-bound structures, is that the membrane of these vesicles is in fact a phospholipid bilayer. And you would all tell me that the fundamental structure of our cell membrane is a phospholipid bilayer. So these phospholipid bilayers can merge with one another. Well, so here's our tyrosine kinase. And one of the really interesting aspects of tyrosine kinases is that, and here's the key word, they auto, you guys are all going to tell me auto means self, they autophosphorylate. They phosphorylate themselves. Where do they phosphorylate themselves? They phosphorylate themselves on the beta subunits where there are these tyrosine residues. Here comes our first messenger. And in this case, The first messenger is insulin. This, I'm now making more specific. We're going to say this is an insulin receptor. So if there was a true-false question or a multiple choice question, and it says something to the effect that the insulin receptor is a second messenger system, it's a receptor-gated ion channel, or it's a tyrosine kinase, you guys would all sit there and say, tyrosine kinase, that's what it is. Everybody with me on that? So insulin comes along and it binds to the alpha subunits of the insulin receptor. And when that happens, that changes the structure of the insulin receptor. And that causes the beta subunits, which possess the kinase properties of this protein, to switch from an off to an on state. So I'm going to say it one more time. When insulin comes along, it binds to the alpha subunits of this insulin receptor. That changes the structure of this kinase, of this protein. And it's the beta subunits that possess the enzymatic or the kinase properties of the insulin receptor. And so what happens? It ends up phosphorylating itself. Where does it phosphorylate itself? On these tyrosine residues within the beta subunit. So here I'm just going to now put PO4 minus, PO4 minus. Now we've phosphorylated these tyrosine residues, and now through a series of events that I'm not going to get into, it's not necessary for you to know this, ultimately the phosphorylation of the tyrosine residue within the insulin receptor ultimately is going to tell these GLUT4 vesicles to fuse with the cell membrane. Pretty cool biology. So let me just restate what I said, just to be clear, is that the binding of insulin to the alpha subunits turns on the kinase properties of the beta subunit. The beta subunit phosphorylates itself. That then, through a sequence of events, which I'm not describing, talks to the vesicle and tells the vesicle, hey, you need to fuse with the cell membrane. Well, using the very simple illustration I have up here, you would tell me that that vesicle possesses 4 GLUT4s, and now those 4 GLUT4s are going to be incorporated into the cell membrane. Now we're into logic 101 land. What's going to happen to the uptake of glucose by this cell? Is it going to stay the same? Is it going to increase, or is it going to decrease? Everybody's going to tell me we're going to increase the uptake of glucose, yeah? No, maybe so? Here's the logic part. What are you gonna tell me is gonna happen to the concentration of glucose outside the cell? It's gonna go down. What's gonna happen to the concentration of glucose in your blood? It's gonna go down, right? Okay, so that's our tyrosine kinase system. I'm using insulin and the insulin receptor as an example. There are other types of tyrosine kinases, but as it so happens, this is part of a bigger story we're building. At this point, all of you should be able to tell me how glucose is metabolized by a cell to make ATP. If you've forgotten it, go back and review it. Now, you should be able to tell me a little bit about how it is that we regulate the uptake of glucose by cells, and that's through this hormone called insulin. And this is going to become part of even bigger picture next week. Let me just pause there and ask if there are any questions, confusion. Don't be bashful to speak up. Yeah.
So, from the tyrosine to the GLUT vesicle, it's binding. So the insulin is basically just binding the phosphorylates to this B subunit, which causes the tyrosine kinase to basically just move to the GLUT vesicle. No, I lied. Ha ha.
So when insulin binds to this insulin receptor, there's the receptor part, those are subunits, and attached to them are these beta subunits. The beta subunits possess the kinase property.
Oh, so the alpha subunit will
Alter the structure of the beta subunits,
yeah, because
right? Because all this story is all about is altering structure, which then alters function.
but it alters it because the insulin binded with the
That's correct. To
alpha?
the receptor, that's correct.
So
yeah.
if you ask me like, okay, what initially causes the alpha to change its structure, I would say the insulin chemical messenger.
Insulin binding to
Or
the alpha
insulin binding
subunit.
to the alpha what? Alpha
Subunits.
subunit. Alpha subunit. And then that causes a change in the structure of the beta subunit. And is the
Uh-huh.
beta subunit a tyrosine kinase?
It is a tyrosine
it is
kinase.
a tyrosine kinase.
Mhm.
And so the tyrosine, what's tyrosine?
It's an amino acid.
It's an amino acid. Okay, yeah. It's an
What
amino
are
acid.
proteins made up of?
Amino acids.
That's right.
Okay.
So it autophosphorylates itself. And we
Okay.
know specifically it doesn't phosphorylate other amino acids, it phosphorylates these tyrosine amino acids. And there actually are many of those, and there's a specific region that becomes phosphorylated.
And we get this tyrosine from the food that we eat, right? When we break it down.
We get it from what?
The food that we eat when we break down.
Well, yeah,
proteins.
ultimately.
Yeah.
Yeah.
That's where we get all our amino acids from. And so is it binded to the tyrosine kinase, our beta subunit? Is it binded to it? And then that, no?
So this is a protein.
Mm-hmm.
Proteins are made up of amino acids. There's a whole bunch of amino acids in the beta subunits. And one type of
And it's made out
amino
of
acid
tyrosine.
in the beta subunit is tyrosine.
Okay. Could I say that it's made up of tyrosine amino acids, or is that wrong?
Could you say that the beta
That the
subunit
beta
is made
sub-
up of tyrosine?
Yes. Amino acids?
I wouldn't say that.
You wouldn't say that?
It's made up of amino acids, right?
But
It's
just
not
not
made up just
just-
of tyrosine,
Okay, so okay,
right? It's
but
a protein.
primary.
There's many different types of amino acids in there. The target amino acid is tyrosine,
Gotcha. Okay.
all right?
Yep, that makes sense now.
So this example, is this the kinase type of transduction, or?
This is a type of transduction, in this case, of using kinases, but specifically tyrosine kinases.
Okay, so number 3 of the 4.
Yeah.
Okay.
And we're interested in these
Right.
tyrosine kinases because this is the category the insulin receptor falls into.
And then the one that we did before with IP3, that is in itself just just the secondary messenger system, or is that considered to be receptor gated ion channels too? Because there's one at the very
No,
top.
so over here, this is a IP3 second messenger system,
And only IP.
because the second messenger is IP3.
Oh,
See, the name given to the second messenger system is due to what the second messenger is. So we have over here a cyclic AMP second messenger system. Over here, our second messenger is IP3, hence
So
it's referred to as
technically,
IP3
when-
second messenger system.
When we start talking about the endoplasmic reticulum, is that then the receptor gated ion channel type of transduction?
Typically, no, so transduction, we're thinking about occurring at the cell membrane.
Okay.
So it just
So-
so happens to contain a receptor-gated ion channel at the end.
And then you'll learn as we delve into the neurobiology, you'll learn about some receptor-gated ion channels located on the membranes of dendrites.
So so the insulin receptor in its entirety is a tyrosine kinase, not
Correct.
just the beta subunits.
That's correct. And it turns out that the beta subunits is where the kinase properties are located. Yeah, go for it.
Do the beta subunits have anything to do with beta cells, or are they separate things that both coincidentally glucose?
Yeah, so if I heard your question right, you said, is use of that term related to beta cells, is that right? Yeah, so you're talking about beta islet cells. Yeah, so those are cells, these are proteins. So when we talk about the beta subunit, we're talking about this proteins, it's got many different parts to it, or regions to it. In this case, there's regions that we're gonna say are alpha subunits, or regions that we're gonna say are beta subunits. Does that make sense?
Yes.
So, this is a molecule, not a cell.
Okay. So they just both kind of coincidentally have to do with glucose regulation.
Yeah, right, it's purely by the way in which individual scientists come along and they label things alpha subunit, beta subunit. It has nothing to do typically with the cell in which those receptors are found, those subunits are found. All right? Other questions? All right. Now, before I move on to nuclear hormone receptors, here's an important point I want to highlight for all of you, is that in each of these instances, where is the chemical messenger acting? Say it loud.
At the cell membrane.
At the cell membrane. And that tells you something, because it's something important about the chemical messenger. In each of these instances, if we're talking about the cyclic AMP second messenger system, IP3, tyrosine kinase, these molecules, these chemical messengers must be what? Are they hydrophilic or hydrophobic? Are they lipophobic or are they lipophilic? They are hydrophilic, right? Because if they're hydrophobic, they would by definition be lipophilic, and they'd be able to move through the lipid portion of the bilayer. So in each of these instances, our chemical messenger is acting at the level of the cell membrane, because our chemical messengers, our first messengers, are all hydrophilic. Really important point.
I know, but in comparison,
Well, so within the cell, here's another type of organelle. I'm just using a circle, and I'm going to label this as the nucleus. So we're talking about something inside the cell. We're not talking about the cell membrane. We're talking about the nucleus. And now I'm going to erase what I'm showing you here. And what I'm going to simply do is just represent this part of the nucleus. Everybody with me on this? So all I'm going to do is we're going to zoom in and we're going to look at what's going on inside the nucleus. So here's our cell membrane. Over here, we're going to say that this is the nuclear envelope. You might sit to say, well, that's the membrane of the nucleus, but I don't want to get into the structure of the nuclear envelope. It's a little bit more complicated than what we typically think of. Within the nucleus, we've got our genes. And this is a grossly simplistic version of what takes place, but I'm just going to come along. This is going to represent a gene. And we're going to say that there are 3 regions in this gene. There's going to be this region here that I'm kind of filling in with this little red squiggly mark. And we're going to refer to this region as where our so-called regulatory response elements are found. So listen to those words, because those are actually pretty powerful words. Regulatory response elements. So here I'm going to put capital R, E. Regulatory response elements. I'm just going to write that here. And these regulatory response elements are the portion of the gene that determine whether the gene's turned on or turned off. And different molecules can bind to different regions within this regulatory response region. There's also, I'm just going to put a little arrow here pointing in this direction. And typically, this is meant to represent what's called the promoter. The promoter promotes, it turns on the process by which we make a mirror image of our DNA in the form of what is known as messenger RNA. So here I'm just going to write promoter. And this promoter can be either turned on or turned off. So let me just back up. We're going to say that the promoter region is what turns on this process known as transcription. Transcription is the process by which we make a mirror image of the gene itself, the coding region of the gene. This region that I've got in blue here, this is what we're going to refer to as the coding region of the gene. This is the region that actually codes for the sequence of amino acids that ultimately will be put together to make a protein. So here I'm going to put coding region. All right, so that's kind of our thumbnail sketch of what's going to take place. So as I mentioned to you a few moments ago, we can have these chemical messengers which are hydrophilic. They're able to dissolve in water and move around in the aqueous environment. But they're also chemical messengers that are not hydrophilic. They are hydrophobic. So how the heck is it that we're able to carry these hydrophobic chemical messengers and take them to the target cell? And to do this, there are so-called carrier proteins. So here I'm just going to write carrier protein. And when these carrier proteins bind to a lipophilic or hydrophobic chemical messenger, that makes that whole system hydrophilic. So here is a steroid, I'm just going to put for purpose of this illustration, a steroid hormone. At the core of steroid hormones is a lipid known as what? Anybody know? What are you saying?
I said steroid.
Yeah, steroid hormone, but at the core of steroids, what makes up the core of steroids? Cholesterol, that's exactly right, very good. So if you follow the cholesterol story, which all of you will, when I get done with you, I'm going to brainwash you, because all of you, we want to become good health citizens. And one of those is one way in which you're going to do that is to try to prevent the development of cardiovascular disease. And at the core of a lot of cardiovascular disease is this bad little molecule called cholesterol. Now, cholesterol comes in good forms and bad forms. There is a type of cholesterol, listen to the term, HDL. What does that stand for, HDL? High density lipoprotein, right? High density. Well, how do I make a fat high density? I attach a protein to it. Okay? And then there's what's called LDLs. Those are the bad forms. You don't want your LDL levels to be above 100 milligrams per deciliter. Low density lipoprotein. There's not much association of protein with it. It's mostly cholesterol. All right, so we've got this carrier protein, and bound to it is a steroid hormone. The steroid hormone is hydrophobic. By binding to the protein, we make it water soluble. And now, through this process of diffusion, this carrier protein that has this steroid hormone bound to it, it comes along and it binds to a docking site. This is kind of cool, that within our membranes there are docking sites. So this comes along and it binds to this docking site. And remember, this is a lipid. This hormone is a lipid. And when you sit there and think about the structure of our cell membranes, the majority of the structure of the cell membrane is phospholipids. So our hormone can diffuse through the lipid portion of the bilayer. But immediately we have a problem as soon as that hormone makes it to the inner surface of the cell membrane, because the most common molecule here is water. So you guys are all going to tell me there's got to be something within the cytoplasm. What is it going to be? A carrier protein. That's exactly right. Very good. Excellent. And through random molecular motion, this is going to make its way on over to the nuclear envelope. And here, through a similar type of mechanism, our steroid hormone is going to, along with this carrier, is going to dock here, and then it's going to diffuse into the nucleus. And the fluid inside the nucleus is known as what? Anybody? Cytoplasm, you would call this the nucleoplasm, right? The nucleoplasm. So the hormone finds its way into the nucleoplasm, and here it has to bind to another protein. And now we're going to give this carrier protein in here a more specific name. And we're going to call that a nuclear hormone receptor. Good term. NHR. So I'm just going to put capital N, capital H, capital R. That stands for nuclear hormone receptor. Had the pleasure of working with an individual down at the Salk Institute. If you don't know where the Salk Institute is, it's down by UC San Diego. You can go down there, and right by the Salk Institute, they have a launch site for hang gliders. Kind of a cool place to go. Anybody ever been down to the hang gliding place on it? If you're ever bored someday, just go on down there and you watch the guys run along, and off the cliff they go. Flying along. And this individual down at the Salk Institute, his name is Ron Evans, he discovered this whole family of proteins called nuclear hormone receptors. And so this is a huge discovery, because we're talking about proteins that are binding these chemical messengers that regulate the expression of genes. Huge, huge story. Okay, now, so I'm showing this is a very minimal region here, but there are many different elements, the elements are simply sequences that can bind to these nuclear hormone receptors. So there's many different types of nuclear hormone receptors. Some of them turn on the promoter, some of them turn off the promoter. But they're very specific, right? We want to regulate the biology of the cell in a very specific, intentional way, not a haphazard way. So what happens is that when our hormone binds to this nuclear hormone receptor, it changes the structure. How many times have I said this so far in this semester? It changes the structure of this protein. And this is important, because if the protein could bind to the regulatory response elements in the absence of the chemical messenger, that's not a very well regulated system, right? We want something that's very highly regulated and very specific to all types of chemical messengers. So when the hormone binds to this nuclear hormone receptor, it changes the structure of this receptor, which will now come along and bind to a very specific region of this regulatory response element. And here I'm just going to show it docked here. And now, by binding here, it's either going to turn on or turn off the promoter. What does a promoter control? It controls this process called transcription. And just very simply, I'm going to describe that as making a mirror image, even though that's not technically correct. But we're going to say we're going to make a mirror image of this coding region of this gene. So when this happens, when the binding of the nuclear hormone receptor binds to its regulatory response element, if it's a positive element, then that's going to turn on this process, this sequence of events that's called transcription. And in so doing, we're going to make a molecule of messenger RNA. And let me be clear, this molecule of messenger RNA, we're going to say, is a mirror image of our gene. And make sure you understand that genes code for proteins. Simple rule, but that's our rule. All right, here's what I'm going to do. I'm going to stop here and let me just give you the summary of this story. This molecule messenger RNA is going to be exported out of the nucleus into the cytoplasm. And waiting inside the cytoplasm is a decoder. The decoder is called a ribosome. And I'm just telling you, our biology is spectacular. The ribosome is going to attach at one end of this messenger RNA, and it's going to walk along the length of the messenger RNA and decode the information in the messenger RNA to end up making and what did you guys learn? What was principle number 3 at the beginning of the course? Genes code for proteins. And what's the machinery of our cells? It's more complicated, but this is our basic mantra. Proteins are the machinery of cells. And if we're making a normal complement of proteins that have a normal structure to them, life's going to be pretty good. If we're not making enough of a protein, then life may not be so good. And of course, this is a problem as we age. As we age, our genes become damaged. And that's a whole nother area of biology that's spectacular, absolutely spectacular. It's that in every one of our cells, in the nucleus, there are gene repair systems. So when a gene gets damaged, boom, the cell comes right along and very, very quickly repairs that molecule of DNA, yeah.
When was this discovered, man?
What's that?
When was this discovered?
The presence of nuclear hormone receptors, this is probably 1980-ish or so, early 80s. Yeah, yeah. And Ron Evans, who, like I said, I worked with him for a little bit. He is a giant in the field of molecular medicine and basically has won a number of prizes. They're like a precursor to the Nobel Prize. Don't know if he'll ever win a Nobel Prize, but he's won some very, very, very prestigious prizes.
This is like almost proof for epigenetics, that, in a way.
Epigenetics is kind of a different
Is it?
phenomenon. Yeah, yeah. This is not epigenetics. But, yeah. Anyways, it's fun being around people like that because these are the truly people who are the great breakthroughs in medicine. Okay, that's it. I'll see you guys upstairs. We'll say in about 7:10ish or so. And what we're going to do is we're going to continue the lab. you started last week, I'm gonna give you a preface to that, and that's the name of the game tonight.